Hey there, ladies and gentlemen, boys and girls. Today I want to talk a little bit further on our first step of genetics, talking a little bit about mathematics. All right, a big part of genetics is, and our study of it is going to be statistics, probability. All right, that's when we talk about inheritance. We're talking a lot of times about the odds of something happening, and a great tool to do that is something called a Punnett square. A Punnett square is nothing more than just a probability table. The odds of the genotype from two parents resulting in this type of genotype, 50%, 100%, 25%. It's all about the odds. Now, the odds are different than what actually happens, but it gives us a chance to kind of predict and see certain patterns. All right, so we're going to kind of go lean back on our information about what we know about alleles, which are different versions of the same trait. Right, we talked about the law of dominance, which is the dominant allele versus recessive allele. We're going to assume right now that there's just one dominant allele, one recessive, just to make life a little bit easier. All right, and then we talked a little bit about what it means to be a homozygous dominant, homozygous recessive, and then heterozygous. If all that seems kind of vague for you, make sure you look back in your notes to kind of remind what that terminology is. So the easiest way, what we're going to do today is we're going to talk about what we call monohybrid crosses. All right, or monohybrid cross. Mono being the prefix for mono being single. All right, we're going to do it for one trait. All right, now this is very uncommon because typically we talked about how it's very complex and there's typically multiple traits working or genes working together. But we're going to talk about this as one trait. Just make this nice and easy. So we're going to pick an example here. Let's pick. We're going to pick something very, very simple. Let's talk about polydactylism. Sounds really hard. It's actually not that hard. Polydactylism is poly meaning many, dactyl meaning like digits. All right. How many digits we have on our hands. And so we're going to talk about the number of fingers a person has. All right. So we have two traits for number of fingers. We can either have 5 or 6. There's two alleles, a dominant allele and a recessive allele. So we're going to talk about the number of fingers. So to represent this, I'm going to use the letter F to represent our genotype. All right, so I'm going to use the letter F here. I'm going to have capital F to represent the dominant allele. I'm going to have a lowercase f to represent the recessive allele. Remember, the law of dominance says that the dominant allele would always show its phenotype over the recessive. Now, actually, what kind of shocks people a little bit here, the dominant phenotype for polydactylism, or the number of fingers you have, the dominant trait is 6 fingers, while the recessive trait is 5 fingers. Now remember, dominant doesn't mean more. All right? With an entire population, each having 2 alleles, there might only be 1 or 2 alleles in the entire population of Dublin that has that dominant allele, which means it's not going to spread as much. All right, because they're going to pass down that gene. If they don't pass down that gene, it might go extinct. Dominant doesn't mean more. It just means it shows its phenotype in a certain circumstance. All right, so there's basically 3 genotypes we can have here. All right, we can have homozygous dominant. Oops. We can have homozygous dominant. We can have heterozygous. Then we can have homozygous recessive. So those are our 3 possible genotypes. And then we talk about, out of those, both of these will result in 6 fingers. And this one here would result in 5 fingers. Right? So here's our genotypes, and here's our phenotypes. All right, so now let's do what we call a cross. A cross is when we basically see what are the possible genetic outcomes from 2 individuals. So let's say we got a mom and a dad, right? So let's start over here. All right, let's say we have, let's say dad, it has 6 fingers. Let's say he's heterozygous. Right? He's heterozygous. He's got 6 fingers, right? He's got 6 fingers. And we know that we are diploid. We get one. He got one from his mom and his dad. right? But during meiosis, remember the purpose of meiosis is to send gametes here where it has half the number of DNA. All right. So eventually he's going to split these two apart, which we'll talk about that in a second. All right. Let's talk about mom here. Mom, let's say she's also heterozygous. Maybe they met on a six-finger forum. All right. So what's going to happen is to have an offspring, they wouldn't give up both of their DNA and both of their alleles, right? Both of their genes and both their genes, because that would be 4, right? We have to give up 2. The whole purpose of meiosis is to break this up. So being a male, what they're going to do is they're going to go through meiosis and create those sperm cells, right? And what they're going to do, we talked about during meiosis 2, is meiosis 1 and meiosis 2 is we're separating these. Right? And mom is going to do the same thing, except she's going to separate hers in the form of eggs. There's her dominant allele there, and here's her recessive allele here. All right? And we want to know what is the possible combination. So what we just did here is we went through meiosis to create our gametes. All right, we went through meiosis to create our gametes. Notice our gametes have only one cell or one version of that allele because they got separated. All right. Now I want to see what are the odds, like what's the possible combinations. If we got to fertilize to make a zygote, we need a sperm and an egg. So do we use this sperm and this egg, or this sperm and this egg, or this sperm and this egg, or this sperm and this egg? We got to find out all the possible combinations. And so the best way to do that is to make a grid. All right. To see all the possible combinations here. All right. So what we're going to do is, now that we have the mom's gametes on one side, dad's gametes on the other side here, we just see, okay, big F, we're going to end up with a homozygous dominant here. Big F, one from mom, or one from dad, one from mom. Going across here. We're using this chart. We always put the dominant one first. So this one here, we always put the dominant allele first. And then little f and little f. This is our Punnett square. All right? This is an example of what we do across here. So what we just did here is we crossed two heterozygous individuals. All right, those were their genotypes. And we ended up with this here. All right, so here are all the possible combinations that we could have. We could have a homozygous dominant, heterozygous, or homozygous recessive. All right, so now we got to talk about what are the possible genotypes and phenotype outcomes of this. All right, so out of all the possible genotypes, I'm just looking at the genotypes here, just seeing like which ones could possibly have been made. So it looks like we always do this we always list this from the most dominant, 2 dominant alleles, to the most recessive, 2 recessive. So out of this one here, I had a ratio. I just don't have a good red marker. All right. Of 1 homozygous dominant to 2 heterozygous to 1 homozygous recessive. And we always do it in that format, all right, where we do the homozygous dominant, heterozygous. So we go from most dominant, kind of least dominant alleles, all right? So another way to write that is we could just write it straight up as 1 to 1 ratio. All right, that's the ratio that we got there for the genotype, or the genotypic ratio. right? Or we could say another way to think about it is just, this is 25%, because there's one out of the four options. One out of the four options was this, 25%, 50%, 25%. All right, that's just genotypes. So we have a 1 to 2 to 1 genotypic ratio for a cross between two heterozygous individuals for the number of fingers. All right, that doesn't tell me the number of phenotypes. All right, now the phenotypic ratio, remember, that's all about what each one of them looks like. What is the outcome, the physical description of each one of them? All right. All right, so now we got to look at each one here. All right, I had, this one would be 6 fingers, right? This one would be 6 fingers. This one would be 6 fingers. That one would be 5 fingers. So I have here just kind of a ratio. We have 3 6-finger, right, compared to 1 5-finger. Or we could rewrite that ratio as 3 to 1 ratio. They have a 3 to 1 phenotypic ratio. You always put the dominant one first, right? Or I could say that there is a 75% chance that these two individuals' kid is going to have 6 fingers. There's a 25% chance they are going to have 5 fingers. Now, they could have 10 kids and all of them have 5 fingers. That's the difference between probability and what actually happens. A purpose of a Punnett square, this is a Punnett square, is to just tell me the odds, given the different genotypes from the parents there. All right? So, kind of going back through, I'm going to kind of go back through this one more time because there's a couple things I want to say. All right? So, remember, we have our dominant allele. We will represent the dot or the big letter. We have our recessive allele. We have the different 3 possible genotypes, which result in different phenotypes. These ones would be 6 fingers because they have the dominant allele. This one would be 5 fingers. All right. Then what we have is we have the cells separate during meiosis. Mendel talked about this as being something called the law of segregation. Segregation, sorry. I still feel like I spelled that wrong. If I did, I apologize. All right, law of segregation says that these alleles split. During meiosis. All right, they separate from each other. All right, once they separate from each other, we can see what are the odds of the different egg and sperm combinations. All right, as we do that. Now, this generation right here, mom and dad, we call this the parent generation. Because this is the ones we started with. And then when we get down here, the possible kids, we call this the filial generation. Filial just means offspring in Latin. It means like kid or next generation. All right? And so what we actually call this the filial generation. And this is the first generation. So this would actually be called the F1 generation. If I take these two here and then cross them, they have kids that would be the 2 F1s. We create an F2 generation, kind of grandkids, if you will. All right. Once we do that, then we have to measure out, okay, what are the number of genotypes, and we put as ratio, we always put dominance first. Most dominant, so we have 25% dominant, homozygous dominant, 50% heterozygous, 25% homozygous recessive for a 1:2:1 genotypic ratio. Now, it's not always going to be the same for the phenotypic ratio. Sometimes it is, and sometimes it isn't. All right? For a phenotypic ratio, we have 3 6-finger outcomes and only 1 5-finger outcome based on the genotypes. Because remember, genotype determines phenotype. So that's a 3-to-1 ratio, or 75%, or 75 to 25%, or 3-to-1 ratio. All right, that was kind of a lot here. It's kind of a long video because there's a lot of vocab. There's a lot going on in this board. All right, it's a lot of little things. All right, if it feels like you're looking at it now and I kind of don't understand what each thing say, then go back to that part and look at it again. All right, you can ask me in class sometime. All right. Otherwise, that's going to do it for right here. You guys are going to be doing a little bit of practice going forward, just giving a cross. If I give you a cross of, say, a homozygous dominant, homozygous recessive, what is the genotypic ratio and what is the phenotypic ratio based on those genes? All right. Other than that, thanks for watching. Walk back over it if you need to, or make sure this is in your notebook. Otherwise, good luck, and may the science be with you. See you.